soils is the largest reservoir of carbon in the forest.
Sure.
The second would be the tree or the forest biomass. One of the big uncertainties that we have is where that carbon in the soil comes from. We know it comes from the litter, the leaves that fall, and the other plant material, but what role does dead wood play in that? Does dead wood contribute to the soil carbon pool? It's an important question because dead wood, as you can see looking around this forest, you can see all this dead wood. Dead wood comprises about 20%, up to 20% of the forest biomass. So knowing where that carbon goes is very important to us. So one of the difficulties in terms of being able to differentiate where soil carbon comes from is that there's no way to tell from a compositional perspective whether that carbon comes from wood or whether it comes from leaves or whether it comes from the understory material. With the FACE experiment, the free air carbon dioxide enrichment experiment, in order to grow trees under elevated CO2 conditions, that CO2 is manufactured. That CO2 has a different carbon signature than CO2 in the air, natural CO2. So trees grown under this elevated CO2 condition have this distinct, what we call isotopic signature. And so what that means is that we can tell if this piece of organic matter came from wood that was grown under elevated CO2 conditions or whether it came from the ambient background. And so in this experiment, this is a very unique experiment that allows us to determine how much of the carbon that we see in the soil underneath these logs actually came from the log itself as opposed to the leaves and other material that's laying across the soil surface here. So this experiment is designed to determine how much carbon, well, determine first if carbon from decomposing logs enters the soil. That's the first question. Second, if so, how much and where does it go and how persistent is it into the soil? When we started this experiment, the science was really equivocal on whether dead organic matter contributed to the soil organic matter pool. And so this experiment was started to assess that question, but in a very large scale, so that we could speak to the issue at a much larger scale than one particular site. Because if we're only studying sites here, somebody would say, well, what about here, what about there? So this experiment is replicated on 8 other sites across the country, representing major bioclimatic zones. And so the Duke Forest is one of 2 sites in the southeastern United States representing the hot, humid, high precipitation zones. And they range across the country to high elevation, cold, lower elevation, almost desert conditions, to rainforests on the Pacific Northwest forest to provide a wide range of conditions. But the experimental setup is identical on each of the 9 sites across the country. In a forest, the dead wood occurs in two forms. It's horizontally, it falls and it lays on the ground, as you can see here. There's a lot of natural dead wood, and oftentimes before a tree dies, or when a tree dies, before it falls over, it's standing straight up, and we call that a snag. And so we were interested in studying that full perspective of wood decomposition, logs that are on the ground versus logs that are standing up. So the standing vertical logs that you see here, the logs that are in the plastic cylinders there, these are logs that are essentially suspended in air to simulate this natural dead or standing snag. The idea here is that we're isolating that log from the soil. So it's emulating a portion of a snag that's, say, 10, 15, 20 feet or higher, into the stand. But the idea is that any organisms that are colonizing it, they have to come in through the air as opposed to coming up through the soil. And in some of the microbial work that we've been doing, we look at the fungal communities that colonize this wood, and indeed we see very distinct fungal communities that colonize the wood that's lying on the ground as opposed to those that are suspended in air. Another big factor in the reason why we have them suspended in such a way that we can also ensure that there's no connection to the ground is that, as we have here, there's a lot of termites, subterranean termites. And we didn't want the subterranean termites to be, colonizing these logs that were these vertical logs, as we call them. And because termites in a dead snag, they can start in the base and crawl up the tree and go up quite high. But by having these collars, it prohibits them or inhibits them from building their channels to go up and colonize the log. So what we have then is we're incubating logs on horizontal, flat on the ground surface, and then we have these logs that are suspended above the surface. This experiment was this wood was produced not only here on the Duke Forest. The wood here is a pine, but we also have two other species that were grown in the Rhinelander experimental forest, and those are aspen and birch. And so we have three species that are involved with this experiment. And there's cohorts of each of those species and laid out in the same manner on this site and as well as the 8 other sites across the country. This experiment now has been running for, let's just say 10 years. We're just a couple months shy of 10 years that this was put out. And so what you're seeing here is the result of wood decomposition after 10 years, and most of those hardwood logs are now gone. In other words, they're part of what we call the forest floor, and there's no evidence of them. In fact, they used to be here and here, and then this is an old, smaller pine log. It's very, very highly decomposed. Another really important point to share is that as I started with the story, we didn't know how deep, we didn't know if there was going to be carbon moving in, and we didn't know how deep. So we pretty quickly find out that yes, it is moving into the soil, but we sampled the soil to a very shallow depth, 10 centimeters. We looked at it in very thin layers. And particularly here in the southeast, we found that indeed the carbon is moving into the soil, and it's moving fairly rapidly. And we saw this at year 6. And so what we're doing now is we're coring down to a half a meter below the surface here. And so what we're seeing then is we can actually see the movement of the carbon that started out here in the log, then came into the surface mineral soil, and now it's getting migrated down deeper into the soil and replaced with other carbon up in the top. So you can see this wave moving down deeper into the soil. A new finding that's going to come, that is coming out of this study, is that we started this thinking that the carbon or organic matter from this log would just go directly beneath it. It would get leached from the log directly beneath the log. But what we're finding is that carbon also comes out quite a ways tangential to the log as well. And so what this means is that there's this carbon is a lot more distributed, and there's a lot more of it than if we were just sampling underneath the log. And so what you see here is this was a vertical log. We sample underneath it, and then we sample at 2 distances away from the log to measure it. And so we sample the forest floor, and then these plugs are where we core down to 50 centimeters. But this is completely new, and so the question is how does this carbon get from the log out here to the side? But I think what it is, it's going to be the fungal mycelia that are where fungi moved in and they're transporting it out, and it's just the fungal necromass associated with it. This is a long-term study. It was a unique study, and so we have some material out here that is just it's being held in reserve. And so if there's other new ideas that come up or somebody wants to take advantage of it, we have that capability. Because we didn't know exactly how things were going to play out when we started this study, We wanted to have basically an insurance policy that we had additional material that could be available for people. So the objective is to determine how much mass has been lost from this log over time. And so we approach it two different ways, just to give us a stronger estimate of that material, of that metric. And one is we weigh the log out here in the field, and then with this sample we adjust the weight to a dry weight basis. Another way we do it is by looking at the change in density of this material. In other words, we know what the density of loblolly pine is, and we know that changes with time as it decomposes. And so we'll take a sample from this and measure the wood density in this material. So we've got samples from this exact tree in our archive from day one. We have samples from before time zero when they were deployed, and then at every sample interval.
Oh, wow.
And we keep an archive in case people want to come back and analyze it for whatever reason. And that's the case with the wood material as well as the soil materials that we're collecting. The FACE experiment was really significant because it grew plants. There was not only forest plants, but there were also agricultural sites and prairie sites that were done where vegetation was actually grown under elevated CO2 conditions. And it ran for a sufficiently long period of time to provide deep understanding of what the forest or vegetation community responses should be expected or are likely to occur in the future. And that information is now, that basic fundamental science has then now been incorporated into models and tools that are being used to assess how vegetative communities are going to respond on into the future. So it's been immensely important, and this experiment is basically the last piece of it. In other words, that was growing the material. This is now decomposing the material. Cool. And so it's literally the end of the story. I will say, this wood is incredibly valuable because it doesn't exist anymore. You can't buy it. You can't do this material, this kind of a study without it. The way we used to try to change the isotopic signature, we would bring in, say, corn leaves and decompose them here. But that's not the same vegetation type. It's completely different kind of organic matter. And so this was a real unique opportunity for this collaboration with the Duke Forest and the Rhinelander Forest to be able to deploy this material. And then with the stability of having these sites installed now on experimental forests where you can ensure the integrity of the experiment. Like I said, we're going into 10 years, whilst things are really decomposed here, the western sites and some of the drier sites, that material is going to be there for 20, 30, maybe 50 years. We don't know. But even this kind of site where although the logs are highly decomposed, in some cases basically part of the forest floor, it still provides an opportunity for us to look at how persistent is this carbon in the soil material. How fast does that carbon move through the material? So the value of the FACE experiment is going to continue certainly for a few more decades with this study.